one of the mechanisms that might explain differences in susceptibility or resistance is the ability of different corals to produce more or less chemical defenses that protect them from microbial pathogens. So that's why we undertook this project. And we started off with the basic assumption that corals do have antimicrobial activity. And there is evidence for this in the literature in other parts of the world. I did my PhD here many years ago looking at chemical defenses in Hawaiian corals, so we knew they had chemical defenses against predators. So a priori, we assumed there were at least some chemical antimicrobial defenses in these corals. And I'll show you the proof of that in a minute. But our specific objectives were to assess differences in antimicrobial activity among the 3 dominant Hawaiian coral genera, which could explain these patterns of differential disease across different coral genera, which we find in the field. Assess patterns of population level variability among these corals, so why it is that you see, even among the same coral species, why do you see differences in disease levels at different sites. And then look at antimicrobial activity and chemical constituents in healthy and diseased corals of the same species to see if you can explain why sometimes you see one coral that's diseased and its nearest neighbor doesn't have the disease, which is, again, a common pattern. So many of you have probably heard the methods repeated ad nauseum, but just very quickly, I mostly want to focus on results. We collected small replicate pieces of corals from 3 common Hawaiian corals, Montipora capitata, Passophora meandrina, and Parietes lobata, from 3 sites around Oahu. We extracted these corals in aqueous solvents, and then we tested the effects of these extracts against several bacterial strains in bacterial growth assays in the lab. And so the bacterial strains that we used 9 different strains. These 4 are known coral pathogens. They're pathogens of corals in other parts of the world. These 2 shown in green, as well as Serratia marcescens, are bacteria that are found in human waste that can make their way into the oceans. They survive in the oceans, and they could potentially become pathogenic to corals and other organisms. And then these 3, as well as Yersinia and Vibrio chaloi, have been identified from the surfaces of Hawaiian corals. So that's the justification for choosing these particular bacteria for this project. So our first objective was to determine whether differences in antimicrobial activity existed among the dominant Hawaiian coral genera. This is basically, this is the percent of extracts from these corals that exhibited inhibitory activity against those bacteria as compared to bacteria grown without extract, so compared to the normal growth of the bacteria. And these error bars for each of these corals represent their effects against the 9 different bacterial strains. So this graph here just shows that all 3 of the corals that we looked at do have high levels of antimicrobial activity. So this breaks it out by coral genera, shown in the different colors, and by the different bacterial strains. And so this is, here you've got Montipora in red, Parietes in blue, and Passophora in green. And basically what you can see is that there is a high level of antimicrobial activity across the board. But there are differences among the genera. For example, against Orantomonas, Montipora had no activity, but it had high levels of activity against several other strains. So we've got variability among the genera, variability among the bacteria as well, indicating that these the antimicrobial activity is highly selective. So they're not just producing a toxin that's going to kill everything, they're producing specific chemical compounds that have effects against specific types of bacteria. The other things to notice is that some bacteria, the corals are pretty well defended against, and it just so happens that some of those turn out to be the potential human pathogens, Clostridium, Serratia, and Yersinia, or whatever that's worth. And then some of the extracts had stimulatory effects. That's what these little hatch marks are. And that's not really unexpected either, because these antimicrobial defenses are, or these extracts consist of a complex of many different types of organic molecules, which serve as really good carbon sources for bacterial growth. And in fact, many bacteria have beneficial effects on corals, so these stimulatory effects may be important in the bacteria, or in the corals, attracting those bacteria and maintaining their natural microbial community. So moving on to objective 2, we were looking at the population-specific variability in these corals to see if that determined why there were differences among sites. And so we compared corals from 3 sites, Waianae, Miley Beach, and Kaneohe Bay. And here you can see the results for Paredes lobata. And basically there are differences among the different sites, but what's more impressive is if you break that out by individual bacteria. For example, against Yersinia, Waianae and Miley Beach corals were highly inhibitory towards Yersinia, but the Kaneohe Bay corals had no effect on Yersinia. That contrasts with the effects on serration, which all of the corals that we collected were inhibitory against serration. You see similar patterns in Passapormi and Drina, except here there's actually a larger difference among the sites in overall antimicrobial activity. So Kaneohe Bay and Miley Beach had much lower levels of antimicrobial activity than Waianae. And again, you do see differences, very impressive differences against some of the bacteria. Waianae, the corals all killed Vibrio coraliticus or inhibited their growth. And at Miley Beach, they stimulated the growth of Vibrio coraliticus. So there is definitely some population-specific variability. Montipora, there's a little bit more consistency among sites. Again, you do see differences against particular bacteria. But here you can also, I mean, this occurred in all the other corals as well, but just to show you an example, there is even intraspecific variability within sites. So this colony inhibited growth of Vibrio choloi. These 3 colonies stimulated growth of Vibrio choloi, and the 5th colony had no effect.